receptors. There's the cell's membrane again, and here is a G protein linked receptor. Visually, G protein linked receptors are pretty easy to identify because they always have 7 hydrophobic membrane spanning domains. If you look at the protein, notice that it snakes back and forth through the membrane 7 times. Each one of those is a hydrophobic region. Now, why do G protein linked receptors have 7 of these membrane spanning domains? I don't know. I don't know if anybody's ever come up with an explanation for it. It's just one of the characteristics of this type of signal molecule receptor. Sometimes you hear them called 7-pass receptors. That's like an alternative name for this type of receptor. But they're usually called G protein linked receptors, and here's why they call them a G protein linked receptor. Actually, I just thought of something. Think for a second. Oh, G is the 7th letter of the alphabet. I never realized that. It's kind of neat, but it's unrelated to the 7 passes of the membrane. The reason why they call it a G protein linked receptor is there's another protein which is not the receptor protein. That's called a G protein, and these G proteins are usually parked, so to speak, right at the bottom of the receptor, like you see there. So this gray thing is a G protein. It's not officially considered part of the receptor, but normally you'll find it sort of stationed down there at the foot of the receptor. And that's why they call it a G protein linked receptor, because it normally hangs out with this G protein at its foot. What is the G protein doing there? Well, the G protein is in fact a relay protein, or to be a little bit more specific, the G protein is in fact the first relay protein, because remember, the first relay protein is the one that gets activated directly by the receptor itself. Relay protein number 1. All right. Now, remember that for the way these signal transduction pathways work, the first relay protein is supposed to be inactive until the signal molecule comes along. But when the signal molecule binds the receptor, that leads to the activation of signal protein, relay protein number 1, which then starts activating the other relay proteins. So we expect the G protein to be inactive most of the time, since there's no signal molecule binding up here yet. Well, to keep itself in an inactive state, the G protein binds an allosteric inhibitor, which is guanidine diphosphate, or GDP. Here's what GDP looks like. It is an RNA nucleotide. If it looks similar to ATP, that's not just luck. ATP is also an RNA nucleotide, but instead of having an A in the nitrogenous base region, it has a guanosine, a G, in the nitrogenous base region. And it only has 2 phosphate groups, so guanosine dinucleotide. Anyway, so this GDP is an allosteric inhibitor of that G protein sitting down there at the bottom of the receptor, which is incidentally why they call it a G protein, because it binds a G nucleotide. Okay, so relay protein number 1 is still in the inactivated state. Along comes the signal molecule and binds to the receptor. Clunk. When the signal molecule binds to the receptor, that causes a slight shift in the structure, in the folding of the G protein linked receptor, and that change in the structure of the G protein linked receptor causes a slight change in the structure of the G protein. And what that results in is the G protein gets rid of the inhibitory GDP that was keeping it inactivated and changes it in for a guanosine triphosphate, which is really the same nucleotide, but now it has 3 phosphates instead of 2 phosphates. So you might say, well, what's the big deal? You know, the molecule gained one more phosphate. Well, as it turns out, GTP is an allosteric activator of the G protein. And so we've achieved the next step. We've now activated the first relay protein. We now have an activated G protein. It's the responsibility of every relay protein, once it's activated, to find the next relay protein and activate that so we can go through the signal transduction cascade. So the activated G protein now has to find relay protein number 2. But here's the constraint that it has to obey. The G protein is a membrane protein. Some of these amino acids are hydrophobic amino acids, so it has to stay in contact with the membrane. It's got to stay anchored to the membrane. So it can't, this G protein cannot leave the membrane and go deep into the cytoplasm to look for relay protein number 2. But what it can do is move sideways through the membrane. Membrane proteins are always allowed to drift sideways around in the membrane. And so that's what it does. It'll now detach itself from the G protein linked receptor and just drift through the hydrophobic membrane looking for relay protein number 2. What they found when studying these G protein linked receptors and the G proteins that are associated with them is that there's 2 families of relay proteins number 2, 2 types of relay protein number 2 that the G protein can activate. One of them is called adenylyl cyclase, and that's not a typo. It actually ends in YL followed by another YL, adenylyl cyclase. And another, in certain other signal transduction pathways involving these G protein linked receptors, there's an enzyme called phospholipase C, which functions as relay protein number 2. So in certain cells, relay protein number 2 is adenylyl cyclase, and in other cells, relay protein number 2 is phospholipase C. Now, both of these are membrane proteins, and which makes sense because the G protein can only move in the membrane, so we kind of expect that relay protein number 2 is going to be a membrane protein. Let's look at the adenylyl cyclase signal transduction pathway. All right, so adenylyl cyclase is a membrane protein. It's essentially the second relay protein in this signal transduction pathway that started with the G protein link receptor. Like all relay proteins, it's normally in its inactive state until it gets activated by relay protein number 1, which is the G protein. Here comes that activated G protein. The G protein activates the adenylyl cyclase protein, like so. And when the adenylyl cyclase protein is activated, it is an enzyme. It now becomes an active enzyme. And what it does is start manufacturing a molecule called cyclic AMP, or CAMP for short. The adenylyl cyclase grabs an ATP molecule and converts that ATP molecule into a cyclic AMP molecule. Oop, go back there for a second. All right, here's the structures. There's good old ATP. What the adenylyl cyclase does is break off these 2 phosphate groups, and the last phosphate group forms a new covalent bond that links back onto the ribose sugar, like that. And so it forms essentially a ring structure that around this phosphate, and that's where it gets its name, cyclic adenosine monophosphate, CAMP for short. So anyway, the adenylyl cyclase enzyme now becomes a factory for these cyclic AMPs. It just keeps grabbing ATP after ATP after ATP and keeps converting them into more and more and more of these adenylyl cyclase molecules, which, as you can see from the diagram, just go drifting down into the cytoplasm. The cyclic AMPs are small hydrophilic molecules, so they do not have to stay with the membrane. They can drift down into the cytoplasm. Now, why on earth is the adenylyl cyclase doing all that? Well, what our goal here, as usual, is to try to activate the next relay protein in the pathway, and that's what those cyclic AMPs are going to do for us. The signal transduction pathways that I've been showing you so far are ones where each relay protein phosphorylates the next relay protein to activate it. But there's another type of way that a relay protein can activate the next one, and that's by releasing these small molecules like the cyclic AMP. The molecules like cyclic AMP are called second messengers, and they're always small non-protein molecules that are allosteric activators of the next relay protein. So the next relay protein is going to be turned on by the cyclic AMPs. Why do they call them second messengers? Well, the people who were first studying these signal transduction pathways said, well, this is the first messenger. The signal molecule itself that's outside the cell is actually the first messenger, and it allows for communication between one cell and the next cell. But these small molecules that drift around inside the cell, like cyclic AMP, are like the second messenger. They're like a signal molecule going from one protein in the cell to another protein in the cell to activate it. All right, so adenylyl cyclase makes the second messenger molecule cAMP. And there you see it. Here's the G protein linked receptor binding its signal molecule. Count them 1, 2, 3, 4. Oh, I left out it's supposed to be 7 passes through the membrane. Looks like I left one out. Okay. There's the activated G protein, which has drifted away from the receptor to go over and activate adenylyl cyclase. And there's adenylyl cyclase releasing those cyclic AMPs down into the cytoplasm. So we have receptor, relay protein 1, relay protein 2, and so now our goal is to activate relay protein number 3. Well, the relay protein number 3 is allosterically activated by the cyclic AMPs. So eventually one of those cyclic AMPs will diffuse on downward to relay protein number 3, and by changing the shape of the protein we'll put it into an active state. And now relay protein number 3 is going to activate relay protein number 4. Now, in what way will relay protein number 3 activate relay protein number 4? Well, a lot of these signal relay proteins are kinases, so at this point what we've done is probably activate a kinase, maybe a tyrosine kinase, maybe a serine threonine kinase, and that's probably how it's going to activate the next relay protein. So just because the signal transduction pathway did not really begin with any kinasing going on, we didn't have any phosphorylation up here really, it doesn't mean that later on down in the signal transduction pathway we can't have some kinasing involved with the relay proteins. Sound alrighty? Let's see something real quickly here. Go back. Oh yeah, okay, I know what I was going to show you. Kind of a subtlety. There we go. All right, so there's the G protein linked receptor, there's the G protein which got activated, and it activated adenylyl cyclase. What happens to the G protein? Well, after the G protein has done its job and activated the adenylyl cyclase, it eventually goes back to its inactivated state. It'll get rid of that GTP that's activating it and change it back into a GDP, which is an inhibitor, and then it goes back over to the receptor that it started at, and eventually just is now inactivated until the next signal molecule comes to that receptor. Alrighty, so after the G protein linked receptor is activated, the G protein, in some cases, the adenylyl cyclase is relay protein number 2 that gets activated by the G protein. In other cells and in other signal transduction pathways, instead of this being the second relay protein, a different protein in the membrane called phospholipase C is the second relay protein that the G protein activates. So let's see that in action. There's the G protein linked receptor, the signal molecule finds it, the G protein gets activated because it now has a GTP that drifts sideways through the membrane and is now going to look for this phospholipase C relay protein. There it is. It's in an inactive state at first because it hasn't encountered the G protein yet. But here comes the G protein. The G protein activates phospholipase C, and phospholipase C starts cranking out a bunch of a type of molecule